So I'm going to consider a question which is related to the aspect of cytoplasmic inheritance, where we study maternal inheritance pattern. And the question is, in a class of Limnia, the snail contributing the egg was dextral. Limnia is a gastropod snail, and in this snail, the shell coiling may be dextral or sinistral, which is actually under the control of genotype as well as the cytoplasmic content. So the female which is contributing X is dextral in phenotype, but it is of unknown genotype. Its genotype is not known. Both the genotype and phenotype of the other snail to which it is crossed are unknown. Okay, all the F1 offspring exhibited dextral coiling. This is another point to consider, that F1 snails, they show dextral coiling. Now 10 of F1 snails, okay, all those snails which were actually F1 types, they were allowed to undergo self-fertilization. You see, this is another important thing to consider, that Limnia is a hermaphrodite snail. It can show cross-fertilization as well as self-fertilization. So it could be crossed with, means male-female crossing could be male. And if a single snail is allowed to produce progeny, then in that situation it will be undergoing self-fertilization process. So F1 snails, uh, 10 of the F1 snails were allowed to undergo self-fertilization, and one half produced only dextrally coiled offspring. Means out of 10, 5 showed dextral coiling in the offspring, whereas the other half produced only sinistrally coiled offspring. So what were the genotype of the original parents? This is the question. And to solve this question, you should know the genotype of the parents which were actually having dextral coiling, but the other parent to which it was crossed, its genotype and phenotype was not known. But F1, all the F1 progeny, they possessed dextral coiling. And these F1 progeny, when they were allowed to undergo self-fertilization, then their 50% of progeny were actually dextral type, and remaining 50% were sinistral type. So with this, we can solve the question. See, I have shown here the answer of this question. Now parents, one of the parents which is actually contributing the X is female. Okay, so that is female. As I just said, that Limnia is a hermaphrodite snail, but if it is laying eggs, then that is being considered as female. So this would have been definitely either of the genotype capital DD or capital D small d. But see, if it is capital D, then they are all F1 progeny will be, they will be heterozygous. If it's both alleles are dominant, then F1 progeny will have to be heterozygous. But we find that the F1 progeny, they are also producing the sinistral type. It means these female parents would have been definitely heterozygous. So the parent will be capital D small d, and capital D allele will help the production of that specific protein which causes dextral coiling. And the other parent, we believe, would have been small d, that is recessive type. So this male would have been, it means the sperm was contributed by a individual, and that would have been homozygous recessive. So all the F1 progeny would have been either of genotype capital D small d or small d, because if you make a cross between heterozygous and homozygous recessive, then there would be 50% heterozygotes capital D small d and 50% small d. So these F1 progeny will definitely show dextral quelling, because even those who have small d genotype, they will be showing dextral quelling because they actually receive cytoplasmic content from the female who is already containing dominant allele. So because of this dominant allele, the protein responsible for dextral quelling that is already present in the oocyte, so they will be dextral in nature. But what will happen if these F1 progeny are allowed to undergo self-fertilization? Then those who would be capital D small d, they will show dextral phenotype in the next generation, and others who are actually small d, although they are also looking dextral in their phenotype, but they will produce progeny only of sinistral type. So it is being said that F2 progeny are all, you know, means they are not showing only dextral phenotype, but 50% of them are dextral and remaining 50% are sinistral. So all those from DD, capital D, small d parents, means heterozygotes, will be dextral, while others from small d parents will be sinistral. So by seeing this, we can say that the parent's genotype would have been capital D small d, and that would have been in case of female, whereas the male parent uh, with which this female was actually crossed possessed genotype small d small d. So this would be the correct answer.